Okay, so let's begin with our introductions. Hello, everyone. Welcome to today's webinar. My name is Marissa Fraser, and I am with Pion Inc. and will be moderating today's event. This webinar is titled Creating Consistency and Predictability in Cannabis-Infused Beverages, and will be presented by Harold Hand, CSO at Vertosa, and we have Deb Shatner, homogenizer specialist from Pion Inc. So as the CSO and co-founder of Vertosa, Dr. Harold Hand spearheads the development of industry-leading customized active ingredients for infused product makers, offering pre-suspended aqueous solutions to create homogeneous, stable products while maximizing bioavailability, clarity, and taste. His work at Vertosa combines his historic background in emulsion chemistry and science, along with curiosity and fascination in the rapidly growing cannabis industry. Developing nano and microemulsions his entire career, Harold holds a PhD in surface chemistry from NYU and is the author of 2 patents in emulsion chemistry. Now, before founding Vertosa, Harold was the senior emulsion scientist at Bio-Rad Laboratories, where his team scaled emulsification oil and supply products to over 90 countries. Now, also joining us today is Deb Shatner with decades of experience at B International, serving in a wide variety of roles, including CEO. Deb has most enjoyed working with the team to bring success to each customer project. Equipment manufacturers are a unique breed where technology, quality, and reliability matter most. Corporate dedication to continuous innovation has driven B high-pressure homogenizers to expand mixing, blending, grinding process capabilities for particle size reduction and cell rupture applications worldwide. Since Pion and B International have combined forces, Deb now serves as homogenization specialist for Pion. Now, we do want this webinar to be interactive and encourage you to contribute to its success by asking questions at any time during the presentation through the Q&A box on your screen or widget. Our speakers will answer them at the end of a presentation, and as your moderator, I will be posting as many as time permits. Now, if we don't get to any questions during the live session, we will cover those via email after the event. If you do need assistance during the live event, use the chat widget. You can just send your questions or concerns to the host, and I'll be monitoring that widget as well, and we'll be able to help you during the presentation. Lastly, I want to let everyone know we are recording this presentation, and we're gonna make it available to you on our website and also via email notification. And now, with further ado, I will pass things over to Harold, who will be sharing his presentation, followed by Deb. Thank you for your attention.
Thank you, Marissa. And thank you, Pion, for offering me this opportunity to present our work at Vertosa to produce the reliable backbone ingredients for our industry. So I will start with a self-introduction. Hi, everyone, I'm Harold. I'm an emulsion chemist. My whole training, my whole knowledge is based on how to stabilize oil into water. And Marissa did a fantastic introduction of myself, my past experience in working in Bio-Rad. And working in Bio-Rad actually introduced me to cannabis because, working in California is legalized. Cannabis was legalized here. And I had an opportunity to try myself. And, when I realized this plant is not making me disengaged or being lazy, which I've been told, but actually is making me happier, more connected, and more focused. I realized, this plant can help society a lot more. That's where I made a decision to wanting to come into the industry, but I didn't know where to start. And when talking to many friends and advisors, combining my background in the emotion science with my love to the plant, this crossroad between cannabis and emotion seems to be the ideal fit for me. So what do I believe? I believe the plant can heal both our body and our mind. And I do believe nowadays the society needs more need from this plant for healing purpose, because we have too much stress and too much technology. That's our goal, to spread happiness and connection to the society. So Vertosa, what do we do? Vertosa produce cannabinoids that is water compatible. We are a B2B company, so that our clients can use our ingredients to infuse into beverages, edibles, cosmetics, and any products contain water. Why do we do it? So we want to unlock the healing power of the cannabis plant and make it approachable and accessible to all. And that is the whole purpose of the whole team at Vertosa. That's why we work so hard every day to deliver this promise. So over the whole cannabis supply chain, we are ideally positioned in the infusion step. So we don't touch any plant. Our starting material begin with distillate or isolate, and we turn those materials into a high concentrated cannabis liquid emulsion. And because of that change, our clients can use this ingredient and to put their imaginations in creating a unique product for the consumers. So Vertosa has been in the business for 5 years. We're located in Berkeley, California, and we are operational in Canada and in other 19 states. So it has been a great experience so far, helping clients launching their products and essentially making millions of consumers experience cannabis beverage, which is a new category. So what do a consumer look for in the cannabis beverage? I think still down to 2 words: consistency and predictability. So today, my presentation will be focusing on those 2 words and present how, at Vertosa's point of view, we help drive the trust of consumer in those 2 words. Consistency. Consistency means consistent potency for us. And to talk about this, we have to share some of the pitfalls that potency can get lost, right? So things you want to avoid to ensure you have a consistent potency. Potency can be lost either physically or chemically. So physically, it can be either have issues with ingredient compatibility or packaging compatibility. On the chemical side, the potency can be lost through thermal processing stability or oxidation on the long shelf life. So I'm gonna give 4 examples given those 4 titles. Let's talk about ingredient compatibility. So when you add cannabis emulsion into a beverage ingredient base, because the beverage can contain lots of different ingredients at variety of different pHs, and sometimes you don't even know what you cannot even predict how would the cannabis emulsion be compatible physically with the beverage base. So what we actually invented is Vertosa, when our clients want to launch any new SKU, we will ask the client send us the new SKU, and we do a physical compatibility for free for 2 weeks. And during this period of time, the results will be pretty obvious to tell which emulsion formula should we use for those SKUs. And of course, we develop different formulations to be customized to fit different needs. So in this case, it's pretty obvious we're gonna select organic 2 formula for this client, for this SKU. So the goal of doing this is to avoid failures when you produce in a mass production in a tank. Packaging compatibility. The best example for this is the can liner. As we know, in the aluminum can, what's holding the liquid is not aluminum, but it is a thin layer of the polymer we call liners. And those liners are hydrophobic, meaning they like oil. And our emulsion droplets inside it is hydrophobic. So hydrophobic, like hydrophobic. So the emulsion droplets tend to stuck to the liner. And this phenomenon is known by the word of scalping. So scalping will cause the potency in the liquid to drop, right? That will reduce the potency. This issue has been a huge problem for our industry 4 or 5 years ago in California. That's why everybody was using glass at that time. So we want to solve this problem for the industry. And the way we solve it is by working with leading can liner companies. And we have request samples from different liners. And as you know, the can liner is actually a very complex engineering issue. They have very different complex chemistry. And then we'll combine the liner chemistry with the emulsion chemistry. Sometimes it's unpredictable. And what will be the combination of the potency stability? So what we do here is we get all the liners, we get all our formulas, and we measure the formula with liner stability. And over a 20 weeks period of time, we create this matrix so that our clients, when they choose the formula, they can ensure if this formula is stable with that certain liner they use. Okay, let's move on to thermal processing stability. So this equipment you see here is called retort. So retort is a very harsh condition to kill the microbial in a can. So why we do retort? Why we do thermal processing? Because a finished or ready to drink beverages, to ensure a shelf life, you need to reduce the microbial load in the beginning to a very low level, and so that stuff doesn't grow in there for the consumer safety. So we have this case study that our client want to make a cold brew coffee, and the cold brew coffee usually has a high pH. So the low pH will inhibit microbial growth. The high pH will usually facilitate microbial growth. So in order to have a long shelf life, those products need to go through a very harsh condition. That's called retort. The condition is about 150 degrees C for 5 minutes. So it's a reduced pressure, but high temperature. So this harsh condition will kill everything every microbial in a drink, but it may also introduce negative effects. For example, we have done 3 SKUs here, and then we see around 10% potency loss on the CBD pre and post retort thermal processing. And then we continue to do stability along 20 weeks timeline. And if you know that certain thermal processing will consume a certain percentage of the potency, then you can overcompensate, right, in the beginning to ensure that consumer in the end get enough potency in their product. The last one for a potency as a risk is oxidation. So inside a beverage container, the oxygen can exist in 2 places, either in the head space or in the dissolved liquid space. So in total, it's called total dissolved oxygen, TPO. So how does oxygen do to the THC? As we know, THC will over time be oxidized into CBN. So oxygen is a big enemy for our infused beverages, and we want to reduce it. Otherwise, consumer will get different psychoactive effect from the product. So how to solve this? Of course, as the cannabis beverage co-packers, we strongly advocate them to control dissolved oxygen to be below 100 ppb, which we think is an ideal level for oxygen control. And then also it can be really helpful if you add an antioxidants as ingredients inside the beverage. So those antioxidants can delay the oxidation reaction. And also, even you have a low DO, even you have antioxidants. If you have a packaging that will constantly ingress oxygen into the liquid, that could be an issue too. So we are doing study right now to understand between PET, glass, can, and different caps, what is the oxygen ingress rate, so that our clients can be aware and adjust their formula and shelf life based on those information. Okay, I talked 4 steps to maintain the potency. As for the consistency part, and then let's assume you can ensure the consistency, but from consistency to the predictability, I think that is the step further to ensure consumer have a repeatable experience every time they drink this can. So our goal at Vertosa is to make the cannabis beverage as predictable as a beer. When we drink a wine or beer, we know how much beer can be dangerous for us, right? We know how soon we can feel the tipsy. But we want to get that experience, reliability to the cannabis beverage. And the first thing to do there to understand this whole experience is start with the PK. So PK means pharmacokinetics. Essentially, it tells you when you take a formulation or a active ingredients or drug, how does your body absorb the drug? So in order to know this, the best way is for a human PK study, which I volunteered a couple years ago at Vertosa. Essentially, we sent people to the lab. We draw blood at 10-0, and then we take either the gummy or the infused beverages, and then at time 5 minutes, 10 minutes, 15 minutes, even for 8 hours. At each time point, we take a little bit of the blood from the testers, and then we'll analyze the blood concentration of the THC and its metabolized 11-hydroxyl THC, THC carboxylic. So this will tell us how fast does the cannabinoids enter the blood. What is the maximum blood concentration of cannabinoids, and what are the overall percentage of cannabinoids that can be truly absorbed into our body. So here is the example that we have done last year on the PK study. We have 6 people study, the same 6 people on day 1 take our emulsion formula, and the same 6 people on another week, on another day, take the distillate formula. And we plot the average of the blood concentration and overlay them together. Now you can see that when consuming our emulsion, the Cmax, which is the maximum concentration, is 2.2 times higher than Cmax from the distillate. And the most interesting thing is the Tmax, the time to the peak. When you take the emulsion, the Tmax is much, much shorter compared to taking the distillate. And even the quicker, for us, it's surprising to find out the Tmax actually is within 10 minutes. In another way, after consuming our emulsion, within 10 minutes, your blood concentration can reach a peak. So that indicates, how efficient our technology would deliver the active ingredients into the circulation, and so that you can have this, instant gratification and the feeling. The purpose is, you avoid consuming too much of the cannabinoids. So both consistency and predictability rely on a great deal on a good cannabis emulsion. And how does our cannabis emulsion look like? So this is a typical fridge at Vertosa. We have different jugs, and they look like milk, but they are super concentrated cannabinoids emulsion. And you can see different colors. That's because we develop different formulas for different clients, depending on their taste, appearance, experience, and label claims. But what I see as an emulsion chemist is inside of each jug, there are billions of those smaller droplets flowing around. So inside each little dots, those are either THC or CBD wrapped around by our emulsifier. And you can see those dots are really universal sized. That's where a good technology combining chemistry and physics will produce this good quality emulsion at high scale. So I'm going to pass down the mic to Dab to talk about how potentially we can make those high quality cannabis emulsions. Thank you.
Thank you, Harold. I have to say, I always learn so much when I'm in the same room with you. It's so exciting. Thank you. So I'm here to talk about our high pressure homogenizers and how we help producers create just the right cannabinoid for their infused products. I'm going to talk about our high pressure homogenization, what we've learned from our cannabis partners, and then I'll go through some case studies that I think are relevant. So who are we? Our name says it all. B stands for Best Emulsifying Equipment. We're experts in high pressure homogenization, for targeted particle size reduction, for producing stable micro and nano emulsions, and for delivering solutions, in this case, to the cannabis industry. We've been providing solutions for over 30 years. And today we are a part of Pion, which has given us worldwide sales and service representation. We turn pressure into possibilities for our customers. If a picture says 1,000 words, let's look at what's here. On the left, you see a particle size distribution chart. And what you see there is, while conventional mixers produce smaller particles when they mix the product together, the broader distribution here means that they're not getting the as homogeneous as a mixture as they might hope for. And as you see, when you move to what we produce with our high pressure homogenizers, we really produce this tighter distribution of smaller particles. That's what we're about. On the right, you see something else that we deliver. Given a sample, in this case, one formulation, you can produce with our options a variety of results. So why is that important? First of all, it takes a lot for product developers to come up with their formulations. They have to do a great deal of work. And then when they mix them together, the variation of results might be better, as you saw in Harold's presentation, for each product. So you want that variation to get what's best for your product.
Welcome to B International, manufacturers of high-pressure homogenization technology that specializes in cell lysis, particle size reduction, and creating stable nanoemulsions and dispersions. Modularly designed, our patented emulsifying cell provides unparalleled control over the three forces involved in high-pressure homogenization: shear, impact, and cavitation, for extensive formulation discovery and process optimization. With operating pressures up to 45,000 psi and multiple flow patterns available, including parallel flow and reverse flow, processing different materials or products with individual characteristics has never been easy. Engineered with scalability in mind, B guarantees a linear scale-up process with consistent results from small-scale R&D to large-scale manufacturing. B technology is the most versatile and customizable high-pressure homogenizer on the market today, and provides ideal processing solutions across multiple different industries. Reach us at www.beei.com to share more about your processing needs and learn how Bee can help. Bee International. We turn pressure into possibilities. Welcome to Bee International.
So that's a bit about our technology. And next, we're going to get into what we've learned from our customer base in the cannabis industry. So what we understand our product developers need and to create their infused products is that they need just the right flavor. Cannabis can add bitterness to product taste. And sometimes, and oftentimes, they need to mask that bitter taste. And in doing so, they might add some harsh ingredients that would cause excessive wear to the equipment. So that's something that we want to overcome. And our solution was to develop a proprietary coating that coats the contact parts and avoids excessive wear. This reduces manufacturing costs in the long run, and it increases reliability of the equipment. Just the right appearance is important as well, especially in beverages. We know that the cannabis additives have to be clear and they shouldn't settle. Emulsions become clear when all particles are smaller than a tenth of a micron. So what we offer is that maximum particle size reduction or particle size reduction to that ideal target particle size for a given product. And the emulsion stability allows for clear liquids to have a longer shelf life. Key in this industry is this balance. And it's the balance between just the right particle size and formulation to give the potency, taste, and bioavailability that the product wants. Particle size in the nanometer range improves a drug's ability to cross the biological barriers. High pressure homogenization is key to achieving the right particle size, the nanoparticles. Decreasing the particle size also increases the dissolution rate, and that results in faster acting and better absorption into the body. Decreasing the particle size also increases the surface area of the particles. And in that way, it can increase the taste of that bitterness. So for people creating infused products, they have to find that right balance, the one that's going to balance the taste, the fast acting that they're looking for, the potency, all together. And in this way, we provide more process options than any other technology out there and equipment, so that they can produce the best results for a given product. That's the key. Also important to anybody in manufacturing is reliability. So we have a number of ways that we protect that reliability. The first is that when you use traditional premixers, and I'll get into this a little bit more, one of the problems is that they can add air into the product. And that air, adding air, can cause wear on the product. So by pre-mixing with our high pressure homogenizers, you eliminate that we don't add that air into the product. The other thing is that we don't rev our engines to full capacity. Our systems are designed with a slower stroke rate to produce less wear over the long run. So it's just another way that we increase reliability and reduce cost. Also, we're an inline product. You saw that in the presentation. Product comes in and it goes out, and all particles undergo the same process. Versus a batch mixer, where you put a blade in, you add the components to mix them together, And you kind of hope and pray that over time, all the particles will receive that same mixing, that maybe they'll all touch the blade. With us going through that same process, there's no moving parts, and all particles go through an orifice as small as 4/100ths of an inch. So they're all undergoing that same process. All of that adds to that reliability. Let's move to some case studies that we've done. The objectives in these case studies are to study and achieve the target particle size, in this case, 150 to 250 nano within one pass. The next study studies stability. What happens? You get these great results. What happens after a year? Let's look at them. The third is that we've run product on our smallest R&D system as well as our production systems to make sure that they get the same results. We study inline premixing. So we eliminate this other equipment. We do premixing on our high pressure homogenizers. What happens? And the final test involves turbo flow. We add our turbo flow option and we see that can we achieve higher capacity? What do we do in these studies? What are the things that we change out in our studies? Well, we vary all the options, most of which come standard with our standard equipment configurations. We are emulsifying flow pattern. We have reverse flow, parallel flow, inline premixing, turbo flow. We vary temperature. We add back pressure and we manipulate that. That's adding additional mixing forces, particularly cavitation. We vary the number and size of the individual components in the emulsifying cell, the reactors. We vary the size and we vary the number. And we vary pressure. It's a well-known fact that increased pressure in the mixing process reduces particle size. So we vary the pressure and we play with that to see how that affects the mix as well. So the first study was to achieve that target particle size of 150 to 290. On the left, you see the graphical results of the particle size, and on the right, you see the actual data. The premix is up there in between 6 and 31 micron. And here you see the deviation. When you're looking at the first number here, it's what 10% of the results look like. 50 is what 50% of the particle size looks like. And then the 90 looks at what 90% of them look like. So here we see the premix, and then within the first pass, we see that the D10 gets you down to 158 micron nano, 158 nano. And as you go down to the 50 and the 90, you see that it gets bigger. It broadens. That's where you see the distribution. The red is the first pass. So you see this is the 10, this is the 50, and this is the 90. So right there, you see that we've achieved reaching 150 to 250 nano in one pass. The second pass, you see that we get down even further, 113 nano, and then we go to 348 nano. Objective achieved. Now let's look at the shelf life. This is a different product. You can see how beautiful the first results are, definitely in that nano range. And then what happens a year later? What's amazing here is that you see that the variation is under 2% after a year. So now let's talk about inline premixing and turbo flow. One of the reasons we take this modular approach to equipment design is so that we can quickly deliver variations on the technology for our customers, creating the solutions that they need. So here what you see is, because we've got this modular approach, what you see is that the same pressure drop here that causes cavitation, because you're going from a very high pressure to a very low pressure, there's a big drop, so that causes cavitation. At the same time, it can cause what's called a Venturi effect, where it sucks in a separate product component. And that's what we do here. Here you've got the traditional reservoir inlet into the emulsifying cell. And here is the nozzle. But right after the nozzle, we add a second reservoir to add product into. So in the inline pre-processing, what we do here is we add one phase, typically the water phase, through the traditional inlet. And then in that second inlet, we add the oil phase. So here we're doing the pre-mixing. And at the end of the first pass, you've got a mixed product. Following up with the turbo flow, what we then do is we add that mixed product into both reservoirs. In that way, we increase the capacity. What are the advantages? Well, as we said, with inline pre-mixing, you are eliminating other equipment. That means you're reducing processing time because you're not moving from one piece of equipment to another. Another benefit is that you're not cleaning, and later on when validation is required, you're not having to validate two pieces of equipment. So right up front, you're saving time and money and costs. The second thing is that traditional pre-mixers can add air into the product. That air is bad for the mixing process, and it can cause wear, damaging the equipment. So by using the inline pre-mixing, you're not adding that air. The advantages of turbo flow are pretty obvious. You're increasing capacity up to 100% with similar results. And what that means is that your equipment costs go down because you're producing more capacity than you would otherwise. Let's look at the numbers. So the first study here, we take the product and we pre-mix it by traditional means, a high shear mixer or a rotary mixer by traditional means. And then we put it through this first pass of the homogenizer. And what we see is we get those great results down to 200 to 368 nanometer. And the capacity of this equipment is 0.7 of a liter per minute. So that's on this given piece of equipment. That's the capacity. Great results. The second pass, there's no turbo flow. And as you see, there's that improvement. So if you need to get down even lower, you've got that ability here. And again, the capacity is that 0.7 of a liter per minute. Next, we took the same product, but we didn't pre-mix it by traditional methods. We pre-mixed it right in the high pressure homogenizer using the inline pre-mixing that I discussed. And there you can see we get very similar results, really close. And we get that same capacity of 0.7 of a liter per minute. Well, next we employ the second pass, we employ that turbo flow. And what do we see happens? You get even better results than up above, slight variations, but even better. But look at what happens. That capacity is doubled. That's what we wanted to see. So here we see that with inline pre-mixing, we get similar results without the traditional equipment. And with turbo flow, we increase capacity and get similar results, increasing capacity up to 100%. Objectives achieved. In summary, what we've learned in the cannabis industry is that the key to achieving these stable micro and nano emulsions is important to all people in infusing products. You want that maximum bioavailability that you get with that tighter distribution of particle size. You want it to be fast acting, which means that you've got to reduce that dissolution rate again with smaller particles. You want to extend that product shelf life. That comes from emulsion stability. You want process efficiency. You want to get rid of equipment that you don't require, and you want to reduce cleaning time and get the fewer number of passes possible. Reliability is key, as it is for all manufacturers. You've got to stand up to those challenging ingredients, and you want to avoid gas infiltration during pre-mixing, and you want to reduce the equipment costs. That means that using the same equipment for higher capacity, and the modular design means that rather than replacing larger product components over time, all equipment needs replacement parts from time to time, but you're reducing the size of them. So rather than that large cell, you're reducing just the smaller part, which costs less. But we like to think that we do more than just build these machines. We develop human relationships, and those relationships allow us to serve our customers better, as we've seen in the cannabis industry, which is more generous with sharing problems and challenges than any other industry I've seen. We're committed to quality, we're experts, dedicated to problem solving with you, and we're aligned with your goals. Your success is ours. So thank you.
Thank you, Harold and Deb, for sharing all that interesting information with us. We do have some time to cover some of the questions from the audience. Again, if you do have a question, just use the Q&A widget. We're here. We'll be raising as many questions as we can in the next 10 minutes or so. So that being said, I will start with the first question. There is a second increase in plasma THC concentration after the Cmax and the drop in emulsion PK study. How to explain that?
Right, I typed the answer there. So it happens a couple of times. So I think it's less likely to be a testing error. And talking to our testing lab partners, they think it's the intrahepatic circulation, meaning the liver will give the blood back to the circulation, so to get a little bump to that concentration. But that's our hypothesis. I'd love to see more research and getting more evidence there. But that's a good question, though.
Okay, next question. Why target particle size is 150 to 250 nanometers? Is it for cannabinoids only? For other substances, other target sizes?
So I think that was one study where that was the goal of that study. But again, every product is different. And as you saw, the next product had even smaller particle size. But that nano range it's that balancing that I spoke about. So I believe that cannabis, although we're treating it different, it's just another drug, right? And so in the pharmaceutical industry, you see that they need target particle size even smaller, down into the hundreds of nanos and below, and larger. So it really depends on what you want that end result to be for your product, whether it's a high-end IV drug or whether it's a cannabis infused beverage. Really varies.
Okay, thanks, Deb. Do you work with terpenes at Vertosa, and what positives are realized with higher levels? I'm not sure if I pronounced that word properly, but
terpenes. Do you work with terpenes at Vertosa? What? Okay, so terpenes are a interesting active because the density of terpenes are lower than cannabinoids, and so it may contribute to emotion instability. And we produce terpenes previously in the company because our clients asked that. However, we're evaluating the terpene markets. We are experts into cannabinoids emulsion, and previously we thought it can be easier to just put whatever technology to the terpene, but terpene has been tricky. The heat stability, the lower density, the stability of the terpene in the emulsion, it can all vary. And so terpene emulsion could be a different product line compared to cannabis emulsion. So any company need to put really focused efforts to making terpene emulsion the best. So bottom line is I don't suggest you just borrow what happened in cannabis to terpenes.
Okay, thank you, Harold. In your PK portion of the presentation, you said the results maxed out in 10 minutes as example. Does this mean the body has absorbed the maximum active ingredient in the bloodstream for the one beverage? Is the rest considered waste or unabsorbed by the consumer?
That's interesting. No, the graph I was showing, the y-axis is the concentration of the THC. So that's where monitors the time concentration change with time. So the concentration reached the peak early on, but that doesn't mean your body not absorbing. So your body keep absorbing. So the picture I showed you is only within an hour. But if you elong to 8 hours, you can see it's like this, but it's keep absorbing. So I won't say you absorb maxed at all the THC, but I will say the THC get into the body really apparently in the early time. And so that correlates to a strong onset, right? A strong psychoactive effect.
Okay, thank you, Harold. Let me see, next question. Why is the industry base of distillate? Different extracts give different effects.
Yeah. That you, or I
Harold?
can answer this. Go
That's
ahead.
the answer then?
No,
well,
I'd leave that to you, Harold.
okay. Well, we work with distillate. We work with isolates. For example, CBG, CBD, CBN. They can come from isolate form, and we also work with water hash or BHO. So different extraction method indeed bring in different nutrients, right? Terpenes, minor cannabinoids. So we develop an SOP to say, okay, can we emulsify them, right? Because different actives may contain plant wax and plant cells. So those materials we cannot emulsify, but anything that material can be clean and dissolvable into our system, we can emulsify. So yes, our technology can emulsify a wide range of cannabis extracts.
Okay, thank you, Harold. This one's for you, Deb. New rules and regulations are being imposed on cannabis processing, and things are trending more towards pharmaceutical requirements. Can you speak more on the pharmaceutical uses of these systems?
Sure. Don't be afraid of the pharmaceutical requirements. You can do it. We've done it for years. That's our main industry. You really need validation packages is a big part of it. And we supply them for, like I said, IV drugs are so stringent in their requirements, security requirements we address with our software. So we're right there from the smaller systems to the largest, not a problem. And it should happen and it will happen. And it's just going to make the industry stronger.
How are you measuring particle size distribution?
We measure it with a Malvern, but our customers use every kind of equipment out there. And what's really a quick story is we had a customer come back with a drug that was in production, and they wanted to do some changes and optimizations, and they sent out the results to 3 different places. They did it with us, they did it with an independent, and they did it with an in-house. And we were right there in the middle. So, you know, from one system to another, it may vary slightly, but the trend is going to be the same.
Okay, thank you, Deb. Can you comment on the number of participants, repeats, and cleansing cycles within your PK study?
So our PK study was done by 6 people. And the same 6 people did the 2 formulas at 2 different days. And our timeline is, we can go a little longer, like 12, 24 hours. We could collect, urines and feces, and blood. But our focus is 8 hours blood work. So yeah, that's 6 people, 8 hours, only blood.
Thank you. Best surfactant or emulsifying ingredients?
Oh, this is interesting. I actually did a poll on LinkedIn asking our network people, what do you think it will be the best or most impactful emulsifiers for cannabis infused beverage? The options are gum acacia, kelaya, sunflower lecithin, and other emulsifiers. And people have different opinions. Actually, I was doing a continuous LinkedIn post on discussing those and introducing those emulsifiers. If you are interested more, check into just thehappychemist.com. So I will have those posts on this website, and feel free to check in those websites.
Great, thank you. Is there no need to heat product before adding to homogenizer?
It depends on the viscosity of the oil. But what I will say is that our equipment is better suited for adding oil than any other equipment because we've got that secondary feed. So if the oil is very viscous and it can't go through the nozzle in the first pass, then it goes through that immediately after the nozzle, it's homogenized. But oftentimes our customers do, depending on the viscosity of the product, do some preheating.
Okay, thanks, Deb. Any ratio for base, extract, carrier, oil, best to use in nano beverages?
Interesting. Well, if I reveal the ratio, I'll be in trouble because we deem our ratio as a Vertosa trade secret. But what I can tell you is, when you change the ratio or ingredient type, you essentially are changing the flavor, experience, and stability, right? And it changes like organically. So I will say it's technology to assess the stability and experience, but it's kind of going to the art wrong when you assess the flavor, right? The sensory is really human palate testing that. So you essentially, a lot of ratio can work, but the best ratio you have to really focus and do a lot of testing on the equipment side and also on the sensory side to give the best formula.
Thank you. We'll cover a couple more questions and we'll have to end our presentation. We're running out of time. So does the way a product is being delivered determine the particle size you want to achieve? For example, you may want a different particle size for an edible, beverage, a powder, sublingual, etc.
Yes, that, or you have any?
I know that. I think you'd answer it better. You are the emulsion expert, and I love that.
Yeah. So for tablets, so that's where we believe not one formula fits all. I can give you 2 extreme examples: the gummy formula versus beverage formula. Beverage is liquid, everything is flowy. Gummy is solid, nothing flows. So in terms of stability, for beverage emulsion, it has high scrutiny because O-ring sedimentation, layer separation, it can be all visual, right? But all those requirements in the gummy is not required because nothing moves in the gummy, right? So that's where at Vertosa we don't design the emulsion ratio first. We see, okay, what are the products we want to infuse? What are the features, how they produce them at high scale? And then work with the product manufacturers and design ingredients that can be fit into their process and best align with their product feature.
And where we come into play is not knowing that science as much as knowing the problem. So somebody will come and they'll say, I want this particle size, but I want it in one pass. That's where our options will help them achieve that.
Great, thank you. One last question. Do we have time? A few more minutes. Would increasing pressure or times of processing lead to a significant decrease on emulsion particle size?
So the answer is yes and no. There is usually what happens is that you're going to hit an endpoint. So let's say that we're going from that 300 micron down to 200. Well, after some products, and not so much in the cannabis industry, I'm thinking more in chemical, you can run that 25 times and then you're going to hit your max of 25 passes. Then you'll hit your maximum particle size reduction for that formulation and its product components. In the cannabis industry, of course, that happens much quicker. Typically, 2 passes will get you, as you saw, lower. Increased pressure, as I said, definitely has the effect of reducing particle size, but that stops at a given point as well. That's why we go up to 45,000 PSI, because that's where we've seen for almost all the products, the benefit of increased pressure stopping. So it really depends on each product. And what we do is we have kind of a layout of a test plan that you're going to find out pretty quickly in R&Ding your product, where the benefits are. And then you get to that quickly. And of course, you can always go back as you change ingredients, but then you move forward with the best mix.
Great, thanks so much. So we've run out of time. I want to thank both Harold and Deb for their time today, for putting all these materials together to present to our audience. Wanted to just mention again that we recorded this presentation and we will make it available within 24 hours or so, maybe less than that. And you'll also receive an email notification with a link to the presentation. If you have any additional questions, feel free to send them my way. I will make sure Harold or Deb get them. If we didn't get to your question, we'll do the same. We'll reply by email. Have a wonderful day, and do check our website for updates on upcoming webinars and events hosted by Pion. Thanks again to our speakers. Have a wonderful day, everyone. Goodbye.
Thank you all.
Later. Bye-bye.
Bye.